So
okay.
Let's start with our last speaker today. So Professor Miguel Ángel Muñoz. He's a professor in physics at the University of Granada, Spain. He's an expert in statistical mechanics, has worked on, among other issues, in an equilibrium phase transition, critical and collective phenomena, and stochastic process. In particular, he helped to develop the theory of self-organized criticality and approaches toward the study of non-equilibrium phenomena, network theory, and complex systems. His research interests span from the fundamental principle of statistical mechanics to interdisciplinary problems in evolutionary biology, theoretical ecology, and system neuroscience. The title of his talk today is Universal and non-universal scaling in brain activity and artificial neural networks. So you can start your talk as you wish.
Okay, thank you, Joaquin. So as everybody else, I have to thank the organizers, but with the difference that the organizers are my friends and are here in Granada with me. But still, I want to thank them. Also, I want to mention Joaquin Marro, who came to Granada 30 years ago, and he was the founder of our research group. Now we are about 10 professors and many PhD students. And he started this Granada Seminar in 1990, so 30-something years ago. This is the 16th edition. And I think, well, no, I have attended all editions and you've talked in a few of them. My very first talk was in a Granada Seminar, I don't know how many years ago. And so I'm really thankful to them for creating the atmosphere, the environment where I grew up, and also for giving me now the opportunity to present our recent work here. So thanks, all my friends. So today I will be talking about brain activity and various different aspects, all of them related to statistical physics and non-equilibrium views. And you will see, I will go step by step. So the first observation I want to point out is that the cortex of humans, not only of humans, all mammals, is always in a state of spontaneous activity. So even if you are doing nothing, you are as relaxed as possible and you go and look at the activity of your neurons, there is something going on. It's not completely silent. Of course, this is known for a long time. I mean, empirically known for a long time since the pioneering work of Berger almost 100 years ago. He was the first to record electroencephalograms and saw these funny, beautiful oscillations. Okay, so this is an enigma. What's the meaning of this spontaneous activity? What's useful for? It consumes a lot of energy, so it has to be useful for something, right? So my interest in this field arise some time ago with the publication of this wonderful seminal paper by the group of Dietmar Plants, Bex and Plants, and they were interested in measuring the activity of neurons. They started doing it in vitro, but then they also did it in vivo. You have here these videos that are courtesy of himself, of Dietmar, and there you see that neurons fire from time to time. Here you are recording local field potentials. It's a mesoscopic local measure of activity. But what they discovered is that the network becomes silent for a while and then some activity starts. So it's like an avalanche. They call them neuronal avalanches. And the funny thing is that if you study their statistics, I mean, the statistic of the sizes and durations of these events, you get this nice power laws which obey finite size scaling. And they look very much like critical properties of a branching process, as I wrote here. So you have power laws and scaling in these experiments. Of course, this triggered a lot of interest among statistical physicists, as you can imagine. I myself has been working on self-organized criticality, so it was very inspiring to see if this had anything to do with earthquakes or whatever, or something much more complicated, as indeed it is. So actually still, this paper is 1 year old. It's a compilation of critical exponents for the size and the duration of avalanches. So it's not very clear if they are really a branching process or if they are something similar, but you look that there is a lot of different experiments, and actually they have been running experiments in different species, man, monkeys, cat, mouse, different brain regions, different experimental techniques with different resolutions, and you see this type of things almost everywhere. So there seems to be something that looks critical at some scales in some regions, or at least, if not critical, at least, let me say, scale invariant. You have some scaling, that's for sure. Also, when you look at the brain at much larger scales, and this is more related to what in our group we have been doing recently, you also have a very nice properties. For example, in this figure here, you see the activity, for example, in this time series, they are measuring the activity by means of magnetic resonance imaging in different locations. And you see that this spontaneous activity, while the subject is doing nothing, is in a resting state, is moving, is doing something. No? There is something occurs. And what's interesting is that some series at different locations, for example, the yellow and the orange, are correlated. So these correspond to different locations in the brain, but this spontaneous activity are correlated. So you actually have some long range correlation between different regions. And the blue ones instead is anti-correlated. So if you take some time window and you compute the correlation or the covariance, let's say, the covariance between different points. So you have here a bunch of regions, here another bunch of regions, and you make this plot to reflect correlations, either larger than one or smaller than one, you get this, but this changes also in time. It's very nice that you have that functional connectivity, which is changing in time. Nice. So at the same time, some other groups in the human connectome projects, among others, they were detecting where the structural connections are. So similarly, you put here many different brain regions, and you put points of different intensity depending on how intensely, with which strength these regions are connected with fibers, with actual fiber connecting them, right? So you have two different things. All this stuff is called connectomics, very fancy. So you have functional connectomics and structural connectomics, right? And the question is, let's try to construct, let's try to construct whole brain models in which at every region of this mesoscopic description of the brain, we put some dynamical model, and this acts on a network, which is the network of connections we have, the structure, and we try to reproduce the function, the dynamical structure of the function. This is a very fascinating topic, and diverse research groups have been working on this. For example, the one of Gustavo Deco, they are experts on this, but the result is that in almost all cases, you get the best possible results if the dynamical model you put here, whatever it is, I don't enter the details, is set to operate close to a critical point, right? Because only when it's critical, you can mimic the rich variability and the many different scales you observe in experiments. So it's also kind of nice that we have experiments showing you a scaling and kind of criticality at microscopic, but also macroscopic scales. Okay, so all these look critical or scale invariant, so what? So people, starting for 30 years ago or something, started to speculate, let me underline the word speculate, about the important functional advantages that you could have if you operate close to criticality. For example, it's obvious that you can have spatiotemporal correlations, that's good, and a large sensitivity, large susceptibility to inputs, that could also be important. You generate a lot of scales, and that's important to have many scales. I mean, you have various possibilities to speculate on the functional advantage of criticality. But also in the context of machine learning, cellular automata, and artificial neural networks, it was conjectured that operating at the edge of chaos, meaning at the edge of criticality, so at the edge of a phase transition, you can have important, very important advantages for information processing. Okay, so if you want more details on this criticality hypothesis, saying that living systems could benefit from operating close to a critical point, you can see this nice seminal paper by Mora and Bialek, and also my own review paper on the subject that they put here shamelessly. Okay, so something that disturbed me for a while is that when you look, for example, at avalanches, people talk about branching processes and the possibility that you branch critically the activity. Instead, if you look at these large scale models, what people talk about are critical points of synchronization transitions. So it's claimed that the brain, in these works that I mentioned by Decoda, that the brain works at the edge of synchronization transition. So I was very confused about that because everybody talks about criticality, but it's different type of criticality. Sometimes about a branching process, a contact process, a synchronization phase transition, some other type of phase transitions. And we wanted to make that clear. And we came up with this theory that we call the Landau-Ginzburg theory of cortex dynamics, which we kind of liked because it's a mesoscopic description of the cortex, and it happens to have a synchronization phase transition that reproduces many of the features I mentioned before quite nicely, but it also has avalanches at criticality. So we kind of put everything together within a unique model. And then there is also this work by the group of Zhu in Hong Kong, and they did something very similar to our approach, but at a microscopic scale. So it's a model of integrate and fire neural, and they have the same thing, synchronization, avalanches, all of this together. But in any case, my personal feeling is that all this criticality thing remains a little bit heterogeneous. I would like to have much more quantitative results, making it able to, to transform this criticality hypothesis in a real theory. We would need it to be falsible, to make a specific prediction and see if they are verified or not. And we are still far from that, okay? We need more experiments and we need more results, and we need also to advance in theory. For example, to advance in theory means that if we have a Ginzburg-Landau theory, I would like to renormalize it. Is that possible? When we submitted this paper and it was eventually published, people asked me, okay, you have the Ginzburg-Landau, now you have to renormalize it, right? It's what we theoreticians like, and I would like to do that. And then, coming from heaven, it arrives this paper 1.5 years ago by the group of Bialek, Bill Bialek in Princeton, which is extremely nice. And they do the following. The thing is that now we have technologies allowing to measure individual neurons at the single cell level. And you can track the activity of many of these neurons, thousands of them, okay? So we can actually have much fine-grained detail on what's going on. The problem is that usually we don't know what are the connections, because the mess of synaptic connections is so complicated that you don't really have an idea of who is connected with whom. So what Bialek tried was something like a Kavanaugh. Here you have Leo and his famous blocks, trying to do something like this, some type of coarse graining, putting together many individual neurons into a super neuron. But rather than grouping them together with the criterion of vicinity, grouping them together with the only thing we know, which is correlation. If you are closely correlated, it's likely you are nearby, and then you will be blocked together. So what they did in Princeton, the group of Bialek, is to analyze a mouse. It's here, it's moving in a virtual maze, and they are recording the activity in the hippocampus. You know, it's the part of the brain responsible for orientation, for location, and these things. Okay, and they were able, look here, they are able to measure less than 1,000 neurons, one at a time. So they record the activity, as you see, for example, in this movie. I don't enter the technological details on this, that escaped to me. But at the end of the day, what they do is this. It's very simple. So you have the activity track of one neuron. This is another neuron, which is the one that it's the closest, the most correlated one to I, okay? So this is the closest one. So we add them up together and we put some normalization. We are going to renormalize, so we put some renormalization to keep the average, to keep constant the total activity. Some normalization. It's the only thing that matters here. And then we have created a super neuron, and we keep on doing this, iterating, so we have these renormalized super neurons, and we can study what's the distribution of activity of these renormalized neurons, right? So this is the distribution of activity at level K, and we recompose it in two parts. This part, P0, is the probability of being silent that we take separately, and this is the probability of non-being silent. And so what's the this depends on X, on the actual value of X. So these are the results, and this is remarkable. First of all, this is the probability of being silent. if the activity was just random, we would expect this straight line here. Instead, it's something else. We have this power line log scale is something that was not expected. This is non-trivial, but let me focus on this one. This is the probability of having some value of X of the activity upon cross-graining. So here you have the collapse of 4 or 5 different levels of renormalization, and you see that they all very nicely collapse into a curve, a distribution, probability distribution, which is not Gaussian. So we have a non-trivial fixed point of the renormalization group. That's very nice. And from that distribution, you can compute the moments, cumulants, whatever you like. Here's the variance. And if you plot the variance here, you see these points which are scaling quite nicely. If they were uncorrelated, they were random, they should go here. If they were perfectly correlated, they should go here, but they go somewhere in between. So you have a non-trivial scaling, non-Gaussian, and a lot of nice things. I also like very much second part of their work, in which they do not real space, but momentum space renormalization group. This is even more fascinating to me. Look, when you have spin model, think of easy model, for example, right? What you do in momentum space renormalization group is that you consider all the modes and then you cut out the slow modes, right? So you integrate out the small modes. But what are the modes? The modes, the Fourier modes, what are they? I mean, we all know what they are, but let me look at them from this perspective. You have the covariance matrix. So you have correlations, 2-point correlations. And when you do the Fourier transform, what you are doing is that actually you are diagonalizing this matrix. And if the system is translation invariant, the diagonalization, I mean, the modes, these modes here, are eigenfunctions, and the eigenvalues are the ones you have in the propagator. The propagator at the critical point is this thing, right? So what I mean with all this is that the modes are distributed as a power law when you are at the critical point. Let's keep this in mind because we will need this. Yes, I mean, we all know the AC model, but let me show it to you. In the order phase, the distribution of modes that we have is one dominant mode and then a lot of noise. This is going to 0. If you're in the disorder phase, it's white noise. So you have essentially all modes. And when you're at the critical point, you have a distribution of modes. I mean, the propagator, which is here, is a power law, right? And this is the power law because the correlations are a power law, and the Fourier transform of correlations power law, it's another power law. Okay, this is kind of trivial for you people in the audience, but it's important to keep in mind. So what happens now with the real neurons of this experiment? They take the pairwise covariance, so the covariance matrix among all possible pairs of neurons you have. You diagonalize this matrix, and you have the modes of this. So you look at the eigenvalues. This is what plot here. These are the eigenvalues of the covariance matrix. And this is the power law. Okay, the range is small, but what is remarkable is that here we have collapse up to 4 different sizes. So you have removed progressively more and more modes, and the curves collapse very nicely. You can also compute here the probability distribution function of activities, and again, you get this non-Gaussian shape. So they repeat the experiments in this hippocampus of mice for 3 different times, and they get that the exponents are kind of universal. I mean, for this one here, mu, that I will be discussing, they get these numbers, which are, I mean, within error bars, they are quite consistent. And these other exponents for the variance and for the probability of being silenced, all of them look rather robust. I mean, not perfectly universal, but within error bars, kind of universal, right? So let me remind you this observation. This is a comparison between the usual analysis that people do in data science and the renormalization group. So people in data science a lot of times do this principal component analysis, right? So you have some points which are, I mean, imagine you have these 2 coordinates, x1, x2, and as a function of time, you have these variable points here, right? So essentially all the variance, all the variability is located in this direction. So if you take the covariance matrix and you diagonalize it, it gives you this principal direction that accumulates most of the variance. And then you have some residual variance here. And if the real space was, I don't know, 25 dimensional, it would mean that the data live in a low dimensional manifold. And doing this principal component analysis, you can have a low dimensional representation of your data, right? But instead, here, we don't have something like this. We do not have that the eigenvalues decrease very quickly, so that the variance in the further modes is irrelevant. No. Here we have a power law, so we have a lot of modes in the tail that are important. If you put here the cumulative distribution of variance, so you keep on accumulating dimensions, dimensionality in your representation space. In the standard case, you converge very quickly, exponentially. Here is 2, here is something else, and then the rest of dimensions are not adding new variance to the cumulative distribution. Instead, if you have here a power law, you have to keep all the modes, because all of them are significant. All of them give you variance. So you cannot just cut them out, as people do in principal component analysis. You have to do RG, as in statistical mechanics. If you remove the low energy modes, the low variance mode, you have to take them into account somehow. And this is the idea of the normalization that has implemented the Alex group. So in summary, we have this very beautiful phenomenological RG approach. And it's very promising, very appealing. So we want to make new things about that. So essentially, I did a summary of Bialec's work. Actually, Bill Bialek was invited to the seminar, to the Granada Seminar. He couldn't make it, so I'm trying to present his results, I think that in a decent way. So what do we do with that? So first of all, I mean, this is kind of summary of what's left. First of all, I will explore whether these results are universal if we go to other regions of the brain, not just the hippocampus. And then in doing this, we discovered a conundrum, a puzzle, something that really strange that I will present to you and then try to solve. And in the last minutes of the talk, I would like to go ahead with a theoretical understanding. Why is all this? Why there is scaling, why it's not? And in particular, try to see if in artificial intelligence, in artificial neural networks, we have similar properties or not. Wait a second and we'll arrive there. So first of all, the first question is this universal? So instead of the hippocampus, you take other regions, you get the same thing? Okay, so we went to look in the open access data of neural activity, Allen Institute and some other places. And we collected collections of individual data sets where you have a lot of neurons, essentially. And here are some of the regions we explored. This one, the olfactive cortex, auditory cortex, primary motor, and here the same type of results I presented before. So this is the variance, and it scales again in non-trivial way for all the different regions with exponent, which is not identical, but similar to the one found by Bialek. And this one is for the probability of being silent, and again, it's pretty similar. The distribution of activities are non-Gaussian and have a very similar form. So my summary, it's here. We have rather good scaling, but with some small dispersion of the values. I wouldn't say this is completely, perfectly universal, right? But still it's very inspiring. But let me go to my favorite exponent. My favorite exponent is one of the correlation of the covariance metrics. Before we saw a decay with 0.7, and here we see 0.8, 0.7, 0.6, 0. It's again with some dispersion kind of universal. So in all cases, we have that the covariance matrix has this fractal, this power law shape in the way that that activity is presented, that correlations in the system are. And importantly, this is very important, all the values are always smaller than 1, okay? So now let me present you my personal conundrum. The conundrum comes from this beautiful paper that I strongly recommend. This comes from the group in the Imperial College of Kenneth Harris. It's a Nature paper 1 year and something ago. And they were registering the activity of 10,000 neurons, one at a time. So it's a new world record. So we want a lot of neurons, and here we have 10,000. We like that. What type of neurons are these? They are looking at the visual cortex of mouse. And what they are doing is they are presenting many different images, natural world images, like 200,000 images. and they record the activity, and then they measure the correlation, the covariance matrix, again, the same thing, the same object of this. And what they observe is this thing that is underlined here in red. They say the evoked population activity was high dimensional, and correlations obeyed an unexpected power law, unexpected. The nth principal component variance is scaled as a power law with 1/n. This scaling was not inherited from the power law of the images. Okay, so what they are seeing is this, which is that the covariance, the spectrum of the covariance matrix is again a power law, but it has an exponent larger than 1. Before in Bialek, it was in all regions smaller than 1. And here is larger than 1 in all the examples they have. But look at this. This is very remarkable. They were able to prove mathematically that the spectrum of covariances, so this spectrum here, needs to have an exponent larger than 1 if you want the representation, the internal representation of the inputs to be continuous, and larger than this number, 1 plus 2 divided the dimensionality of the input, to be differentiable. First of all, look at if the dimension of the input is large, if you have very complex images, this number is very large, so this is very close to 1 in all cases. So what's the meaning of this need of continuity and differentiability? So imagine this is a real world, and you have two different inputs that are nearby, and this is an abstract representation of the large dimensional space where the neural activity lives, and you want to represent these things. So you want these two trees to lie nearby. So you need this to be continuous. So if a small perturbation goes to something nearby, and same thing for this fish. So if it's not continuous or it's not differentiable, then you run into problems. Like, for example, the one you see in artificial intelligence, that you identify this correctly as a cat, but you put this perturbation, which is very small, and then everything fails because your representation is not as nice as the one you have in the brain. It's not continuous and differentiable, and people can play against these networks this way, these adversarial attacks or perturbations. So these are the results from the group of Kenneth Harris. Look, that this is quite remarkable. This is an image which is very complex, and this is a spectrum of the covariance matrix. It's similar but not identical to the one of the actual image, because the image, of course, can be decomposed into components. So it's 1.1, here it's 1.04. But if you put lower dimensional images, you get larger dimensional representations, and this is perfectly according to the theory I mentioned before. You have a lower bound, and the lower bound depends on the dimensionality of the input. So it works very, very nicely, right? And so if you have high dimensional data, you have exponents that are close to 1, but always larger than 1. So now we can apply the RG. The RG told us there is a lot of universality. So we go to do RG with this large set of neurons. Ready? So here are the results. This is the scaling for the variance. Wonderful scaling, even better than before, because here we have 10,000 neurons. And the one for the probability of being silenced, also wonderful. The same type of non-Gaussian scaling, the very wonderful collapse doing either real space or momentum space renormalization group. So everything fits very nicely. So it's in the same universality class of Bialek's thing. But now we have a conundrum. This is the problem. Now when we go and look to the covariance, the spectrum of the covariance matrix, we see rather nice scaling, but the exponent is not the one of Bialek. The one of Bialek is here, smaller than 1, 0.708, and here is larger than 1. If we look at the data for natural images with a large dimensionality, we see exponent close to 1, and if it's simple images, it's larger, and we reproduce that in our RG approach. So we are happy with that. But how can the exponent mu measure in the RG approach in orbiting regions are smaller than 1? And Kenneth Harris is telling us that you need it to be larger than 1 for the representation of the external world to be meaningful. Okay, possible answers that the hippocampus and the other regions we look at operate in a different way as the virtual cortex. That's a possibility. Maybe they are representing information in different way. But what we realize is that, what we realize is the following. No, let me stay here. What we realize is that the data that they have for the visual cortex are not the raw data, so the actual data. They have been filtered. So what's this filtering? The filter is that they have removed trial to trial variability. So they repeat 2 times the same experiment. They see what's the variability in one experiment and the other one. And they are able, with some technique, which is called cross-validated PCA, to separate what's the variability coming from the input and what's the variability coming from the internal noise. And they have removed this one. So we want it back. So we went to the original data. We were able to track down the original data of Kenneth Harris, not the one they had in the paper, but the original raw data. And we did the RG with this one. Again, we get universality, but now for the correlation matrix, we got the same result at Bialek. We have this 0.7 something, okay? So it's like when you have the noise, when you have all the information, the representation is not continuous, not differentiable. You have to subtract noise to really see discontinuity and differentiability of your actual representation. And we did something else. Given that Kenneth Harris filtered out noise, we say the opposite. Okay, now we are going to filter out input-related activity, and we just look at the noise, the spontaneous noise of the brain, right? And again, we have very similar results. And now the exponent is much smaller than before. So if we were able to look just noise in the absence of input representation, we could get a much lower value for this exponent of the eigenspectrum of the covariance matrix, right? So if you are confused, because I'm confused myself with all these numbers, let me plot this here, these numbers here. So if you have white noise, this mu would be 1, so all modes there. If you just have noise, it's 0.5. This is the raw data for brain almost everywhere, is around here. And if you remove noise and you just look, the input representation is this one. And then in the vertical direction is the representation of inputs varying with the complexity, right? So this thing decreases with the complexity of the input, and also decreases here when you remove the input and just leave the noise. So I would love to have deeper understanding of all this. And actually, this is our research program for next month, year, I don't know. So in the rest of the talk, I would like to discuss 2 things in an ideal world. Number one is the approach of a theoretician that would be asking, for example, what's the minimal model that you can build where you can see all these things? So you have this power law of the covariance matrices in the spectrum of the covariance matrices. For example, if you take our own Landau-Ginzburg theory and you do the renormalization group, even if it's phenomenological, you do it in the computer, you reproduce the same numbers? Or more generally, what's the minimal model to do that? There is a very interesting proposal by this group of Nemmenmann, but I will not discuss this here because I don't have a clear idea. So what I will do in the rest of the talk is something else. What I will do is to look for an artificial neural network that we are going, let me go to the next slide. So this is my goal now. What we want to do is to design an artificial neural network. We are going to train it following the standard protocols. It's not that we are inventing anything. We are just following the standard things. And we will train it to recognize and classify images. Which images? The same ones we have from the work of Kenneth Harris and co-authors. And we are going to look inside this network and see how is the internal representation. So we will look how the artificial neurons correlate, what's the covariance matrix, and what's the spectrum of this covariance matrix. It's close to 1. So is the representation continuous, differentiable, it's working nicely, or is this mess? What it is? Let's go ahead. Okay, so this is a very abstract sketch of what we will do. So we will follow this paradigm of artificial intelligence, which is reservoir computing. It's illustrated here. So the reservoir is a network where you have recurrent connections, and they are set randomly, okay? with some random weights. And here is an input layer. So this will be our image. And these guys give an input to all or some of the individual neurons here. And then there is an output layer. So from each or some of these guys, you go to these output layer neurons, okay, and produce some output. And if this output, like to you, for example, if this tells you 1, 0, 0, 0, meaning that this image is a cat, then you are okay. But if it's not, you just have to tune, to change these weights here. So the weights of the output layer. And this is the great advantage of this reservoir computing. So you have here your reservoir, but you are not going to touch the weights of the connections here. So it's not like Hebbian learning, it's something else. So this renders this very, very flexible paradigm for computation. And I personally like this because it reminds me more closely of criticality. Here, what you do is that you project your inputs into some high dimensional space, because this is going to be very complicated dynamics. So you project into a very complicated high dimensional space from which then you will read out whatever, and you will train the readout to be effective. Okay, so this is the very abstract representation. Now we have to adapt this very abstract representation to our actual problem. And here my collaborator and PhD student, Guillermo Barrios Morales, did a wonderful job. I have to say that I would have not accomplished this without him. He's our local expert in artificial neural networks. So he was actually the one doing the hard work. And we took the images, the ones we have on the Harris paper, we projected them in columns into the reservoir, and then we learn, and then we train it to recognize if this is a cat or an owl or something else. Let me just emphasize that we have two control parameters here. This is important. One controls, here, this is the state of a neuron as a function of the state of the previous time of all neurons and the inputs, right? So the things that you have to set are the weights of the internal connections here, and these are controlled by just one number, which is the spectral radius of the connectivity matrix here. So essentially this tells you if the weights are very high, then this is going to be very chaotic if you want, and if they are very low, then the connections are smaller and it's more stable like. And so this is one parameter, and the other parameter is how you weight the input. You can weight it a lot or a little. So these two parameters together, so how much you weight the external inputs and how much you weight the internal connections, control the dynamical state of your reservoir, of your network. And look at this. This is really wonderful, I think. Okay, so we show all the images to our reservoir, and we measure the correlations, the covariance matrix. Now, we are plotting the exponent, this mu exponent that we have been talking about of the covariance matrix. So when it takes the value 1, we represent it with this magenta plane here. Okay, so we see that actually for some particular values of rho and epsilon of my tuning parameters, I have good representations. Okay, are those ones here. So what are the other colors? What's the green and what the red? This is the maximum Lyapunov exponent of the system. So what this is telling me is that, and the critical line, so where the Lyapunov exponent changes sign is here, so it's also the magenta plane. So what this is telling me, I think it's remarkable, is that when my system is unstable, I have positive overall Lyapunov exponent, my exponent, the exponent of the covariance matrix is larger than 1. While if my network is stable, negative Lyapunov exponent, I have an exponent which is smaller than 1. But the thing that's remarkable is that I get an exponent close to unity. So it's the limit, the theoretical limit for continuity and differentiability of representations when I operate at the critical point. Okay, when I am at the edge of chaos, as some people call it, when I operate at the edge of chaos, so right in between stability and instability, I get this value of mu equals 1 predicted theoretically by Kenneth Harris as the optimal for input representations. But look at this. These are one of the images in the sample of Kenneth Harris. This is the spectrum of the image. This one, of course, is much simpler. It has only a few, 7, 8 principal components. And in blue, in orange, you see the empirical results in the visual cortex of the mice that Kenneth Harris et al. Recorded. This is one. And the blue one is the one recorded by our artificial intelligence when it operates at the edge of chaos at criticality. And look that it works rather well for all instances. So this power law that we are seeing here is not inherited from the statistics of the image. It's something else. It's coming from the internal representation that the neural network is doing. We did also a couple of other variants that cover here. So actually we did a noisy version of our reservoir computing machine where we add noise and then we can denoise it using the same protocol of Harris and company et al. And we also get very, very similar results. But look at this final image, which is also kind of remarkable, I think. So, sorry, here I'm changing one of the control parameters that I have. And I'm showing in orange the Lyapunov exponent, okay? So it crosses 0 around here. Here is positive, here is negative. And at the same time, I'm showing you this mu exponent, so the one of the spectrum of the covariance matrix, and it's also crossing 1 in this case, roughly at the same place, okay? So the representation is becoming continuous and differentiable when you enter the regime where you have stability in your reservoir, right? But now this is the performance. In blue, you have the performance, the actual performance of the neural network when it's solving the task. To get this, we didn't work with all the set of natural images of Kenneth Harris because it's too complicated and we wanted something much easier. So we took this data set, the MNIST database, to recognize numbers, and we can do that easily. So actually we have a network with only 500 neurons. So it's a piece of cake, very, very little. And with these only 500 neurons, we are able to reproduce, to learn all these numbers 98% of the times, right? But this efficiency is this high only when we are at the edge of chaos. We are close to criticality. If we go far from criticality, the performance drops very quickly, okay? So the conclusion is that if you want to have a nice representation of the data, which is continuous, differentiable, and works fine, you have to operate close to the edge of criticality. So I think I'm kind of done. Let me summarize some of the ideas that I presented. So number one, that we have spontaneous and evoked activity in our brains. Also mice and monkeys do have similar activity. And understanding it and distinguishing what's spontaneous and what's evoked remains a very challenging task, an important task. So the criticality hypothesis, saying that you have something critical there boiling, blah, 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 is very appealing. And I personally like it, but sometimes, some days I wake up believing that has some truth. Some days I'm more skeptical. So we need more data and more advances in theory to convert it in a falsifiable theory. Then we have this phenomenological RG approach that shows that there is non-Gaussian and non-trivial scaling in real neurons. And this actual evidence, empirical evidence, so we like it, we appreciate it very much. And the results are rather universal across varying regions. We also saw that you can have continuous and differentiable input representations, but these occur on top of a much noisier state. And I was very surprised to see that we could reproduce this in a very simple artificial intelligence model that works extremely well. I have to say, I was really surprised about that. So the big question that remains is to understand if all these features that I have discussed are really an implication of the fact that you need criticality, or there are simpler alternative explanations, okay? So I think I stop here, and thanks you again for your attention and for attending here, for being here at Friday at these late hours. So thanks again.
Thanks a lot, Miguel Ángel, for your very nice talk. It was very interesting for me. So we have now time for some questions. So people interested to ask can do it. Okay. So who is the first one? Is Juan Mazo. You can ask.
Hi, thanks for the talk. Very nice and beautiful. So I was very curious and interested about these similarities between artificial intelligence and behavior of the real neural networks. No, so talking about this, we can always distinguish between structure and function. I don't know if at the end if we look at function, we are doing similar things, but if we look at the structure of the artificial networks and the natural ones are very different. So can you comment something about this, please?
Yeah, well, the network represented, of course, is very simple. It's just random network with some connections. If you want to reach state of the art in artificial intelligence, you have to do something different. For example, you have deep learning, and there you have a lot of layers, right? And in the visual cortex in particular, which is the one that Harris was looking at, you also have those layers, right? So you have a similar structure. Of course, the learning algorithm has nothing to do. The brain does not use backpropagation, but you have some structure. So yeah, I don't know what I should answer you. I mean, if you look at the larger scale networks, of course, they are very complicated. They have modular structure, hierarchical modular, they have hubs, they have a core peripheral structure, they have many different things. And this just enriches the repertoire of possibilities that you can have. So I would say that what we are doing here is something very local. I mean, at the level of local modular computing, something very specific, and there may be this approximation of a random network may work, but of course the complexity of the brain is much larger, as everybody agrees. Right?
We have more questions. Sari Kossi, you can ask your question.
Can you hear me?
Yes.
Yes. Okay. Very nice. Very interesting. I'm a biologist. I used to work in the zone of behavior, but more experimental, not regarding modeling behavior and sex, sexual development and immune system. But I used to read something about this because I used to work modeling some simple systems earlier. One comment is excellent talk, of course, but I would like to point out that recently appeared a paper in Nature describing that once thought one-directional neurotransmission in the hippocampus is now proved to be bidirectional, and with certain qualities like modifying the gain of the signal and on the spread of the signal. Are you taking into account this result? I think it's a paper from this year, from May, from the beginning of May. I couldn't read it because with COVID.
No, I am not aware of that paper. And actually, we are not modeling hippocampus. We are just modeling a very general network, and the one we trained was for the visual cortex, just in case. So we trained it with images. We did not attempt to do anything with the hippocampus. Just look at the statistics of the activity of neurons. So we don't know where it comes from, if it has a symmetric, asymmetric, or if there are placed cells, as actually they are in the hippocampus, we don't have a clue of what's going on. We just analyze the activity, the statistics of the activity. Yeah.
Okay, the comment was directed to the comprehension of that mu with under one value you mentioned in the initial part of the talk.
Yes.
The noise, perhaps what we interpret as noise, might be some kind of signals of modulatory signals that couldn't be taken into account when those data were obtained. Just that.
Sure. There are a lot of sources of noise in the brain. Of course, it's working at finite temperature, and there are millions of things that are controlled by finite number of molecules, a few proteins doing this and that, and a few molecules. So of course, there are a lot of fluctuations and noise is there. So we are not surprised to see it. What is surprising is that in this environment with all this noise, the brain is actually able to store information in a robust and reproducible way. That's amazing part of it, right?
Okay, that's noise is useful. Thanks very much.
Thanks.
Next question, you hi too. Go ahead.
Hey, Miguel, that was a great talk.
Hello, Yuhai. Nice to see you.
How are you? Yeah, so I guess I have a question related to both the AI, the connection to artificial neural network, as well as what happened in the real brain. So I guess these statistics are done when at the resting state, when it's not carrying out any task.
No. All the statistics are done. Well, the two examples we have are the hippocampus and the mice. The mouse is moving in a maze, virtual maze, so it's actually following a path and getting oriented or disoriented. And this is the first one by Bialek, and the second one by Kenneth Harris. They are watching the images, so it's in a continuous set of images. So they are actually representing something. It's not spontaneous activity.
Okay, so can you say something about the comparison? If you are comparing the statistics of the firing pattern versus in two situations, one doing the task versus the resting state, do you see any significant differences?
Well, the thing is that we don't really have data for resting state, because for some reason they always put the mouse to do something. We have not seen
mouse.
Exactly. We have not seen data for the resting state of the mouse. So I would love to have them. I would love to. So the only proxy we have is the trick we invented, that it's take two replica whenever we have two repetitions of the same experiment, and with this cross-validated PCA, try to remove the part from the input. And then we see some residual that we suppose comes from noise and from other ongoing activity, not necessarily noise, but maybe inputs from other parts of the brain. I don't know.
Right. Okay. So the sort of the connection to artificial neural network, I think the connection is certainly some connection is certainly there, but I think in neural network, what people have been mostly interested in understanding is actually not maybe mistakenly, not the firing rate itself,
sure,
neuron, but actually the connectivity. How do you actually learn it? How do you learn a pattern, a representation by adjusting these synaptic weights, right?
Sure.
Sort of the other side of the coin. But this is an interesting question that people probably should take a look. Maybe you have of the actual firing rate statistics and the artificial neural network to see how does it connect with dynamic.
Our approach is very naive, I have to admit. I mean, we just have some empirical data with some statistics. And as this data come from visual cortex, we said, okay, let's do the simplest possible machine learning with visual learning, I mean, visual image representation, and see what happens. And I would have never said that we would get the same type of things if you operate at criticality. So it's kind of nice, but I'm not sure how much this is teaching us about how the brain is actually working, because our network is strange. It's a reservoir computing. We are not changing the weights of the actual network connections. We don't have plasticity. internal plasticity. So that's the meaning of my last question here. So it's really criticality, the explanation for the real phenomenology of the brain, or it's just kind of a coincidence. So it's the best way we can have to reproduce something similar to the brain is by constructing a model which happens to be critical. But I'm not sure that our artificial neural network teach us really anything about the real brain.
Okay, thanks.
Okay, next question, Professor Carver.
Sorry, getting myself unmuted. Excellent talk, Miguel. I really enjoyed it. My question is about this neural network that you were training and was optimally functioning close to criticality. The images on which it was trained, which were this set of numbers, have a certain degree of continuity of patterns. I was wondering if anybody has looked at whether if you try to distinguish between things that say are fractals but have different fractal dimensionalities, so that the images are very discontinuous from one to the next, whether you would see something similar.
Right. Actually, we started from that because this type of neural networks, this reservoir computing, they were proposed to predict time series. So the input is a time series, and you predict it. And for chaotic ones, it works very, very nicely. So you can predict, you get the set of the time series, and then you can predict another set. So the first thing I said is, okay, let's try to reproduce this time series. But if it's chaotic, then it's fractal. So the representation itself, it cannot be continuous because the set is not. So we run into this problem in which if the image or the input is too complicated, is too fractal, then you don't know what you have to look for, because the very notion of continuity and differentiability of the representation kind of break down. So that's why we move to regular images which are not fractal. They have a structure, but they are not fractal. Or we just did this problem, which is much simpler.
Thank you.
You're welcome.
Okay, if there are no more questions, I have a small question for you, Miguel Ángel.
Yeah.
So in your model, in your neural network, your neurons are intrinsically chaotic, or just chaos emerge due to connections because you have asymmetric connections?
The neurons are I mean, not binary, take a value between 0 and 1. So they're analogical. So it's just a number between 0 and 1, and then you connect them recurrently here. And of course, if the connections are very strong, you get a collectively chaotic behavior. Okay.
Yes, because there is a very, very important work from many years ago that show in this kind of stochastic neural network that is a necessary condition to achieve chaos in a neural network, that connections are asymmetric. So they prove it's
here they are symmetric for sure.
Okay.
I mean, we do not impose symmetry.
Okay. And then in the real data, you have noisy activity. So in principle, because there are controversy if the activity in the brain is chaotic or is just stochastic. So how this controversy?
Now we don't know. We just say that here this is deterministic, but we also had a version with noise here, and we get essentially the same results. Actually, we are now exploring whether with noise we can improve the results of the deterministic. The ones I show you here, this performance is for the deterministic case. Now we are trying to see if with a little bit of noise we improve it. Probably we can.
Okay. Okay. There is another question from Gesa Odor. Okay, just the last question.
Hi, can you hear me?
Yeah, Gesa. Nice to
Hi,
hear.
nice to see you, and thank you for the interesting talk. I just have a question about this relation of the exponent nu, which you said a kind of critical exponent, and the beginning you were talking about the avalanche exponents. Is there any scaling relations between them or among them, and then you can predict by observing nu something for these tau, these avalanche exponents?
The answer is maybe. So for example, for the AC model, we know what's the spectrum of the eigenvalue. So we know that this decay exponent here is related to the exponent eta in critical phenomena. So for example, this mu here, if this was the contact process, the directed percolation universality class, of course, depends on dimension. In 2 dimensions, this should be 0.6 in 2 dimensions, okay? So there is a relation between this and a standard critical phenomena. And if you put more exponents, of course, you have relationships. But we are not looking for avalanches here.
But you can also say something about the tau exponents or things like that.
Well, maybe if we had all the data of the actual exponents where neurons are firing, we could do that. But what we have is just matrices of correlations, of covariances. So with those data, we cannot. If we had the raw data with all the timing of the spikes, of course, we could look for avalanches.
Okay, thank you very much.
Thank you, Geza.
Okay, so thanks again to Professor Muñoz for his very interesting talk. And with this, finish our today session of the Granada Seminar. So next section will be on Monday, next Monday. So thanks a lot for all attending people. See you on Monday.
Bye, thanks.